So today's topic is expressed sequence tag. Gene expression is process in which the information of a gene is used to produce a product. That means that information is used for the synthesis of a protein. So next we can say that, so how this particular gene is turned on, then that particular gene will be turned on, and where that gene will be turned on to carry out the function. So that is known as the gene expression. So earlier, if we have to find a gene, then that time scientists were start their search by using the scientific literature. Then they isolate that particular protein, then determine the function of that particular protein and locate the gene that codes for the protein. So that was the procedure, or alternatively, linkage study were used. So nowadays, express sequence tag, this technique is available for the quick searching of a new gene. So this express sequence tag, this terminology was given in 1991 by Anthony Karlevich. The length of this DNA sequence is from 200 to 500 nucleotide bases long. So these are the short single pass sequence that is from mRNA molecule. And these bits of DNA sequence that represents the gene expressed in single cells, tissues, or organs from different organisms. So these express sequence tags are the small piece of DNA sequence. Then these are the short sequences. These are the single pass sequence, and these are read from the mRNA. These are the bits of DNA sequence that represent the gene expressed in the single cells, or the gene which is expressed in the tissues or in any of the organ from the different organisms. So if we see the principle of express sequence tag, these express sequence tags are used to sequence the bits of DNA that represents expressed genes in certain cells which are present in any of the organism, and use these tags to find out a gene from the chromosomal DNA by matching the base pairs. So these are the express sequence tags, are the tags. So these tags are used to find out a gene from the chromosomal DNA by matching the base pairs. So these base pairs are matched, and from these the gene has been found out. So next are the steps which comes under tag preparation. So how these express sequence tags are prepared. So it is the conversion of mRNA into the cDNA, because cDNA is more stable as compared to the mRNA molecule, and express DNA sequences only. So here the only express DNA sequences are present. So first mRNA molecule is isolated. So because mRNA that codes for a particular protein, so the information from the mRNA molecule is used for the synthesis of a protein. So mRNA, this mRNA is converted into the cDNA. Why this mRNA is converted into the cDNA? Because these mRNAs are unstable. So if we have to isolate the RNA molecule from any of the tissue, we have to take extreme care for the isolation of mRNA, because in our hands the RNAs are present, so that while isolating the RNA, it may degrade that particular RNA. So care has to be taken, like wearing the gloves and all that. So this mRNA are unstable, so that's why we have to convert this mRNA into the cDNA. This cDNA are more stable, and these cDNAs are express DNA sequences only. So here you can see the express sequence tag preparation procedure. So this is the mRNA molecule ACUG. In RNA molecule uracil is present. instead of thymine. So after isolating the mRNA from any of the cells or tissues or organs, we have to convert this mRNA molecule into the cDNA. So that means we have to carry out the reverse transcription. So after converting this into the cDNA, then after polymerization, we will get the double-stranded DNA molecule. So here you can see that this is the double-stranded DNA molecule. This is the cDNA, double-stranded cDNA molecule. Then after getting this particular cDNA, we have to sequence that particular ends of a cDNA molecule. This is the 5' end and this is the 3' end. So we will sequence this particular cDNA molecule starting from any of the end 5' or 3' up to how many pairs we will sequence from 200 to 500 base pairs only we will sequence. We will not sequence whole cDNA molecule. So we will sequence these bits of the cDNA sequence. So here this particular green arrow represents how much portion we have sequenced. And this again the sequence which we have sequenced from the 3' end. So this is known as the 5' EST because we are sequencing from the 5' ends. So this particular portion, it is known as the 5' ESTs. And again, if we are sequencing this particular cDNA molecule from the 3' end, then it can be considered as 3' ESTs. So here we are not sequencing the whole cDNA molecule. So cDNA to express sequence tags. So 5' EST sequencing only the beginning portion of a cDNA molecule. That means we will sequence only the 200 to 500 nucleotide sequence of this is that particular cDNA molecule. Because these codes for a protein, this region tend to conserve and not change within a gene family. Why we will sequence only this particular portion? Because this particular 5' and 5' EST code for a protein, and this particular region it will be conserved and it will not change within a gene family. Then if we sequence the cDNA portion on the 3' end, then it is known as the 3' EST. So sequencing only the ending portion of a cDNA molecule is known as the 3' EST. These 3' ESTs are less conserved because it is untranslated region. So if we see the translation direction, it is from 5' to 3'. So these 3' ESTs are less conserved because it is the untranslated region. Next is the uses of ESTs. So these express sequence tags are used to identify unknown gene and map their position in a genome. So by using this express sequence tag, we can identify the unknown gene and we can also map where that particular gene is located into the genome by using the express sequence tag. Then this express sequence tag provide simple and inexpensive path for discovering a new gene. So if we see the procedure of this express sequence tag, we just have to isolate the mRNA molecule and synthesize the cDNA molecule from that mRNA molecule. Then we have to sequence the bits of that particular cDNA, not complete cDNA molecule. So this is a very simple procedure and it is also inexpensive because we are not sequencing the whole cDNA molecule. Then these express sequence tag are used in genome map construction. So by using this express sequence tag, we can construct the genome map. Then next, these express sequence tags are the used for the characterization of expressed gene. So by using this express sequence tag, we can find out which is the expressed gene present over there. Then these are the fast and cheap technology. This is the fast and cheap technique. Almost all the steps are automated. then they represent the most expensive available survey of the transcribed portion of the genes. So by using express sequence tag, we can find out which genes are expressed or transcribed in that particular genome. Then these are indispensable for gene structure prediction, gene discovery, and genome mapping. So these express sequence tags provide experimental evidence for the positions of exons. So by using this express sequence tag, we can get the position of the exons. That means exons means the coding region and introns means non-coding region. So from this express sequence tag, we can get the experimental evidence. From this, we can find out where is the position of the exons in the genome. Then these express sequence tag provide regions coding for potentially new proteins. So which region codes for the new proteins? This also we can find out from the express sequence tags. Then we can characterize the splice variants and alternative polyadenylation. So using express sequence tag, we can find out the splice variants. Where is the where that variants are present and what are the alternatives of polyadenylation? Where is that particular variation is there? So this we can find out from the express sequence tag. Then also this provide an alternative to library screening. So instead of library screening, we can use the express sequence tag and we can discover the gene, then we can map that particular genome. Then short tag can lead to a cDNA clone. So whatever the sequence we are getting, that we can consider as a short tag. So this can lead to a cDNA clone. So we can also prepare the clones of that particular tag. So again, this cDNA, express sequence tag provide an alternative to full-length cDNA sequencing. So if we sequence whole cDNA molecule, then it will be expensive. So instead of sequencing whole cDNA molecule, we are only sequencing the portion of that particular cDNA from either 5' end or 3' end. Then sequences of multiple express sequence tags can reconstitute a full-length cDNA. So if we take the multiple express sequence tag and arrange them, then we can, it may be possible that we can get the full-length cDNA molecule. Then single nucleotide polymorphism, that means SNP data, can be mining. So we can also use this SNP mining by using the ESTs. And next, these are the gene mapping tool, express sequence tags. These are work as a genome marker because these express sequence tags are unique to a particular, because we are extracting that particular mRNA molecule from either cells or tissues or organs. So these are the unique to a particular. Then these express sequence tags are easily recognizable and occur only once in a genome or chromosome. So we can easily find out the express sequence tag and its occurrence only once in that particular genome, or we can say in the chromosome. So that's why it can be used as a genome marker. Then next, these express sequence tags are used for gene discovery. That means they are the resource of gene discovery. This express sequence tag is a tool for the detection of hereditary disease involving a gene. So there are certain diseases which are related to that particular gene. So this type of hereditary diseases we can also find out by using these express sequence tags. Then generation of express sequence tags is rapid and inexpensive. So that's why these are used in the gene discovery. Then next is express sequence tags and NCBI. So in 1992, scientists at NCBI, they have developed a new database designed to serve as a collection point for express sequence tag. So NCBI, they have developed a database that is only for the collection of expressed sequence tag. Once express sequence tag that was submitted to the gene bank had been screened and annotated, and it was then deposited in this new database that is known as the database EST. This particular database regarding the EST is known as the database DB EST, and in this EST database, the only EST sequence is submitted to this particular gene bank, and these are then annotated and then deposited. Then EST databases. The data sources for clustering can be in-house, then proprietary, public database, or a hybrid of these. Chromatographs or sequence files. So each EST must have the following information. As we are hello. This NCBI is maintaining that particular EST database, so that each EST database or each EST must have the following information. First is the sequence ID. Each EST will have a sequence ID. For example, run ID. Then next is the location in respect of the poly A, where the poly A are present. So 3' or 5'. Then next is the clone ID, from which the express sequence tag has been generated. Clone ID is mentioned in that particular EST database. Then organism, from which organism this express sequence tag has been produced. Then from that particular organism, which tissue have been used or under what conditions they have been used. So this information is mentioned. The sequence, the EST can be stored in the FASTA format. Then the format for this particular EST is the FASTA format. So here you can see the example, that particular ID is mentioned over here. Then the organism from which that EST have been prepared. Then from which cells have been taken, that is mentioned over there, and which end end has been used for the sequencing, 3' end or 5' end. Its location has been mentioned in this particular information. Then here you can see that particular sequence of this express sequence tag. So these are the this is the sequence of human endothelium cells, and this is the sequence of 5' end. So here, if you visit the NCBI, then you will get this such type of EST format. Then next are the limitations of expressed sequence tag. So it is very difficult to isolate the mRNA from some tissues and cell types. So there are some types of tissues or some types of the cells from which it is very difficult to isolate the mRNA molecule. So as it is difficult, so we cannot prepare the cDNA molecule, and we can't prepare the express sequence tag for that particular gene. So this is the limitation. Then next is no indication as to the gene from which it is derived. So one of the limitation is it is not mentioned over there to the gene from which it is derived. So this is again the second limitation of this express sequence tag. Next is how to organize express sequence tag collection. So there are 4 steps: clustering, assembling, mapping, and interpreting. So in clustering, associate individual express sequence tag sequence with unique transcript or gene. Then in assembling, derive the consensus sequence from overlapping ESTs belonging to the same cluster. Then in case of mapping, associate the ESTs with exons in genomic sequences. And fourth, interpreting, find the correct coding region. So by using this, we can organize the express sequence tag collection. Then there are certain private express sequence databases are also available. ScanBI, that is from the South Africa. Then MIPS, Munich SIB. Then TIGM, it is from the Italy. Then DOTS from the Pennsylvania. So it was about the express sequence tag.